PCR stands for Polymerase Chain Reaction, and it's a technique we can use to make lots and lots of copies of DNA in these little strip tubes in a machine like this, which is called a thermocycler. So your strip goes in here and it goes down, and then the machine makes it go up and down and up and down and up and down in temperature, as we'll talk about, and that at the end you'll end up with lots and lots of copies of DNA. So let's take a look.
Just review what DNA is. And so DNA is stands for DNA deoxynucleotides. So they're these like building blocks of DNA, and the important thing here is that they have two ends. So they have like a 5' end or position where they have phosphate groups, and then a 3' end where they have OH groups, and they connect through those groups, and that allows them to link up into long chains. And then so that's through the generic backbone part of the DNA. But they also have unique bases, or nucleobases. So this is our A, T, G, and C. And those kind of stick off from the backbone of the DNA like a, kind of like an arm. And these specifically form interactions we call base pairs with other bases. But it's only A to T and G to C. So if you know the sequence of one strand of DNA, then you can copy the second strand of DNA. So in double-stranded DNA, the strands are the complements of one another. And so if you make a copy of the first strand, so you make the complementary copy, then if you make the complementary copy of that strand, you get the original strand. So basically you only need one strand of the DNA to make lots of copies of the whole double-stranded DNA. So and the other important thing to note, though, is that the reason why it's a complement, like each strand is kind of like a reverse complement of the other, because we talk about DNA as being directional. So going from the 5' end to the 3' end is usually how we write things. So this is going to come into play when you design primers. So now let's look at the basics of the reaction. So it's PCR chain reaction, but I like to think of it as a PCR train reaction. So if we think of those strands of DNA as railroad tracks, and they're crossing the continent, and we want to take a journey from 2 points and copy in between. So then we think of our copier, DNA polymerase, as a sort of train, and it can only travel along double-stranded track. So it has to lay down. If you have single-stranded track, it has to lay down the track ahead of it. So as it travels along, it's single-stranded DNA, it's adding the complementary base, so it's forming the complementary strand as it goes. So, but right now you have double-stranded DNA. So in order to get access to the single-stranded DNA to copy, you have to melt. So basically you literally heat up the DNA. When molecules are heated up, they have more freedom to move around, and they like to be free, so they'll wiggle apart from each other. And once they do that, then the DNA polymerase has a chance to get on. But it can only start from double-stranded DNA. So it can only travel along single-stranded when it lays ahead, like the track ahead of it, to get double-stranded DNA. So you need to make double-stranded DNA that's followed by single-stranded DNA. So basically you design short pieces of DNA called primers. And these oligonucleotides, so oligo meaning few, so these primers, you want one to match the start and one to match the finish, but you put one on each strand, because so DNA polymerase, it can only travel from 5' to 3', which is that whole directionality thing we were talking about before. So you need to have one for each strand. So one strand is going to start where you want to start, and then the other strand is going to finish where you want it to finish, and so they like face each other and they copy the region in between. But basically once you have so after you melt it, then the DNA strands are come apart, you have to cool it down a little. in the anneal step. And that's where the primers can get a chance to bind, because before it was too hot, so the primers wouldn't want to bind. But you cool it down a little, and then you let the polymerase go at it, making lots of copies. And so the polymerase needs a little help. So in addition to just having the polymerase and the DNA template in your little tube, you also have those primers we talked about. DNTPs, so the DNA letters, as well as magnesium and salts to keep the polymerase happy and working. And so what's going to happen when you do this is that, so the first cycle is a little different, because at this point, your template is kind of like an undefined length. So you're only going to copy a portion of it. So it's like you're only copying the track from Sacramento to New York. But you need to tell it where Sacramento and New York are. So those primers declare the start stage and end stations. Well, it's the start for each, but they're on separate strands, and the strands are reverse complements. So it's like you have a start and a stop, even though the primers form the start site for the polymerase both times. So in the first cycle, the DNA polymerase is just going to copy till it runs off the DNA or runs out of time or energy or whatever. But so now you have like two, your pieces are different lengths at their end, but they have those, they start at or end at the part you want. They start at the point you want for each strand. And then when that strand gets used as the template, then in the next cycle, you will get it to also end where the part, so then you end where the part that the first primer, that the other primer started at, because the template ends there when you're talking about the other strands. And so then at this point it's more simple, because you have these pieces of defined length that you want, and so you can just make lots of copies of it between them. So yeah, so this is just what I was talking about with the primer design. You want to make sure that it's the reverse complement. So you can find these graphics on my site for reference. And when you're designing those primers, you want to, you're usually about like 20 nucleotides long. You also want to keep an eye on the GC content. So G and C, they form 3 pairs to each other, and so they form a stronger base pair than A and T. And so this amount of GC you have is going to correspond to how tightly the primers bind and like how hot you have to get it to melt it off. So you want to make sure that you have your primers, like they have similar melting temperatures, or TMs. So that's like the how hot you're going to have to get it to like get them to bind or unbind. So you want them to be similar so that you can set the settings of the thermal cycler to one setting that will work for both of them. You also want to make sure that your primers don't bind to copies of themselves, which we call like a primer dimer, so either to itself or to the other primer. And it can also like fold back on itself and form these hairpins. So those are just going to make it harder to bind. And you also want to make sure it only binds where you want it to. So these are fairly short sequences, so you want to make sure that there aren't any even close matches anywhere else in the DNA, or you're going to have non-specific products formed, because the primers will bind there and start, the DNA polymerase will start there, and then you'll get products of a lot of different sizes. So when that happens, you might want to try raising the anneal temp. And so, yeah, happy copying.